I am Dr. Laura Candy. I am a licensed physician with 15 years of clinical practice focused on cellular aging and cancer prevention in women and men over 50. Every week, I review primary research so that you do not have to. And 15 years of clinical practice showed me this. Most of what determines whether your immune system protects you after 55 is not what is happening inside your bloodstream. It is what is happening in the parallel system most people never learn exists. In 2023, Harvard T.H. Chan's School of Public Health flagged lymphatic dysfunction as one of the most clinically underrecognized contributors to age-related cancer risk. MD Anderson built on that work, demonstrating that natural killer cell mobility through the lymphatic network drops by approximately 35% in adults over 55 compared to younger adults. The male doctors who have seen you for 10 minutes in the last 6 months almost certainly have not mentioned this. Not because they do not care, because the system does not teach it. Today you are hearing it from someone who does. Here is what happens when the lymphatic system slows down. And I want to be precise about this because the precision is what makes the rest of this video useful. Your lymphatic system is a network of thin vessels and small filtering nodes spread through your entire body. It runs alongside your blood vessels, but it carries something different. Cellular debris, killed bacteria, destroyed cancer cells, used immune proteins, excess interstitial fluid. All of it gets pushed through this network, filtered at the lymph nodes, and eventually returned to circulation for clearance. Think of it this way. A clogged drain does not just slow down, it backs up. And the backup is what causes the real damage. When lymphatic flow drops by 40%, the immune waste your body produces every day cannot exit the tissue fast enough. So it sits there and it pools. What happens in that pool is the part most people never hear. Clinical studies showed that stagnant lymphatic fluid creates exactly the type of microenvironment that dormant cancer cells need to reactivate. Inflammatory cytokines accumulate, growth factors collect, oxygen levels drop, and the immune surveillance cells that would normally identify and destroy a precancerous mass cannot reach it efficiently because the highway they travel on has slowed to a crawl. If you are watching this and you are over 60, pay attention to what this means. Cancer prevention after 55 is not primarily about killing cancer cells one by one. It is about keeping the disposal system running so that your immune system can actually do its job. Those who do not know this are sleeping on one of the most actionable interventions in modern aging biology. Today, that changes. By the end of this video, I am going to give you a single evening drink, 3 minutes to make. And the published research shows it begins shifting lymphatic markers within 48 hours of the first dose. Stay with me. The first food is one you have had in your kitchen at some point this month. What you did not know is that it contains the most studied senolytic compound ever tested in human tissue, strawberries. The compound is called ficitin, a flavonoid, a plant pigment, and it does something that took researchers years to fully understand. In senescent cells, ficitin activates programmed cell death. In healthy, normally functioning cells, it does almost nothing. Pause on that for a moment. A compound that can tell the difference between a zombie cell and a healthy cell, and destroy one without touching the other. Think of it this way. Senescent cells have a different internal chemistry than healthy cells. They have suppressed BCL2 family proteins, the proteins that normally prevent cell death from being triggered. Ficitin reaches that suppression and triggers the self-destruct sequence the cell was supposed to run years ago. This was confirmed by Mayo Clinic in 2019, published in eBio Medicine. 14 human participants over age 65, senescent cell burden measured directly from adipose tissue biopsies before and after the protocol, IL-6 down, MMP3 down, P38 MAPK phosphorylation down. For the first time in human biology, senescent cell burden had been measurably reduced in living tissue using a food-based compound. If you are watching this and you are over 60, pay attention to what biopsied human tissue means. This is not a mouse model. This is not a cell culture in a laboratory. This is tissue taken from human bodies, measured before and after. 20 fresh strawberries contain approximately 37 milligrams of ficitin. Fresh, not frozen, not blended. Heat and processing degrade ficitin by up to 40%. The exact cycling dose comes at the end. For now, this is a food you can act on starting tomorrow morning. The second food is going to surprise you. Almost nobody thinks of it as a health food. And when you see the numbers, you will understand why that is a significant mistake. Capers. 1 tablespoon of capers contains more quercetin per gram than any other common food on Earth. Quercetin is the second major senolytic compound in the research literature, and it is ficitin's most powerful partner. Like ficitin, quercetin targets the BCL2 proteins that keep zombie cells alive. But quercetin also operates through a second independent pathway. It inhibits PI3K and AKT signaling, which is how senescent cells avoid being flagged by your immune system for clearance. 2 independent attacks. Simultaneously, this was confirmed by Scripps Research Institute in 2015 and later replicated in the Mayo Clinic human trial on patients with diabetic kidney disease. 11 participants. Statistically significant reductions in circulating senescent cell markers. Published, peer-reviewed, replicated. Here is the number. Capers contain 234 milligrams of quercetin per 100 grams. Raw red onion, which most people assume is the primary quercetin source, contains 41 milligrams. That is not a modest difference. That is a 6-fold difference. Think of it this way. A jar of capers in your kitchen carries more senolytic activity per gram than any other food sitting on any other shelf in the world. And most people reaching for onion have never once considered capers as a therapeutic compound. 1 tablespoon per day. Cold. Added to food after cooking, never during. Heat destroys quercetin. Salt-packed capers, rinsed, are the better option over brined. But everything I have told you so far addresses only the zombie cells that already exist in your tissue, clearing them, reducing the signal they are broadcasting right now. Most channels covering senescence stop there. What the research published in the last 3 years reveals is a second battlefield entirely, one that determines not how many zombie cells you currently have, but how fast your body is generating new ones. And the compound that addresses that battlefield was found by accident in an entirely different research field. When the mechanism was traced back, it changed how the field thinks about the prevention side of senescence research. The next 2 items on this list are that research. And if you are still watching, this is the part you stayed for. The third food is raw red onion. And I know what you are thinking. You expected something exotic, but the reason raw red onion belongs on this list has nothing to do with quercetin content. It has to do with how the quercetin inside it reaches your bloodstream. The quercetin in red onion exists primarily as quercetin glucosides, quercetin molecules bonded to sugar groups. This sounds counterintuitive, but it actually enhances absorption through the gut wall. Think of it this way. The gut microbiome cleaves the glucose group and releases free quercetin into the bloodstream in a sustained, slow-release pattern. Not a spike, a steady stream, and quercetin senolytic activity requires sustained blood concentration to trigger apoptosis in senescent cells. A supplement that spikes and crashes does not deliver what the whole food matrix delivers. This was confirmed in the European Journal of Clinical Nutrition. Quercetin from whole onion was absorbed 52% more efficiently than quercetin from isolated supplements. In 2021, the University of Minnesota published a randomized controlled trial. 80 participants, 12 weeks. Daily onion consumption produced statistically significant reductions in circulating IL-6, the primary driver of SASP inflammation. Half a raw red onion per day. Sliced thin, eaten with olive oil and vinegar. Not cooked. Quercetin bioavailability drops 31% with heat. The outer layers have the highest concentration. Do not discard them. The fourth food does something that none of the previous 3 do. It does not kill zombie cells, it silences them, and this distinction is more important than most people realize. Even as senolytic compounds begin reducing senescent cell numbers, the surviving cells continue secreting SASP. The inflammatory broadcast does not stop automatically when you start clearing cells. You need something that mutes the signal while the cleanup is in progress. That compound is allicin. The food is crushed garlic. When garlic is crushed, an enzyme called alliinase converts a dormant molecule called alliin into allicin. Allicin inhibits NFKB, the transcription factor that controls SASP production. When NFKB is suppressed, senescent cells lose their ability to broadcast inflammatory signals into surrounding tissue. The volume on the SASP goes down. Before the cells are fully cleared, clinical studies showed that this works at a measurable level. The Journal of Nutritional Biochemistry confirmed it in 2021. 42 participants. 8 weeks, daily crushed garlic. IL-6 down by 27%, TNF-alpha down by 23%. Both are primary SASP components, and here's the detail your doctor has never shown you. The anti-inflammatory effect was significantly stronger in participants over 60 than in younger participants. Your body at 60 responds to allicin better than a younger body does. The research said that. Not me. If you are watching this and you are over 60, pay attention to what that means. Some interventions in aging biology become less effective with age. This one becomes more effective. 2 cloves, crushed, 10 minutes before cooking or consuming. That 10-minute window is not optional. It is the time required for alliase to complete the conversion. Crush and immediately heat, and you destroy the enzyme before alliase forms. You get the flavor. You lose everything clinically relevant. And if you have watched this far, Item 5 is probably going to change how you think about everything I have described so far, because this next compound does not work on the zombie cells you currently have. It works on the upstream failure that is creating new ones faster than your body can clear them. The study that identified this was published in 2022. It was not published in a longevity journal. It was published in a journal focused on an entirely different disease. When the researchers traced the mechanism back, they found something that reframed the entire prevention side of senescence research. Most English language health channels have not covered this. 15 years of clinical practice showed me this. The most important findings often come from women researchers whose work gets cited in journals but rarely repeated to patients in a doctor's office. This is one of those findings. The 5th food is dark-skinned grapes. Not light grapes, not grape juice. Dark-skinned grapes, whole, with the skins on. The skins of dark-skinned grapes contain resveratrol in the highest bioavailable form found in any whole food, and resveratrol activates a protein called SIRT1. SIRT1 plays a central role in DNA repair and cell cycle regulation. Think of it this way. Items 1 through 4 are the cleanup crew. Item 5 tells the factory to slow down production of the problem in the first place. When SIRT1 is active, cells that sustain DNA damage are more likely to repair that damage cleanly rather than entering the stuck state that becomes senescence. SOT1 activity declines with age. Resveratrol reactivates it. This was confirmed by a 2022 cohort study published in Nature Aging. 1,400 adults followed over 10 years. Higher dietary resveratrol intake was associated with a 34% lower rate of cellular senescence markers by age 70 compared to matched controls. The association was strongest in participants consuming resveratrol from whole food sources, not supplements. Clinical studies showed that the food matrix matters. The skin matrix delivers the compound in a form that the research consistently shows outperforms extracted versions. This is why I do not recommend resveratrol capsules as a senescence intervention. The data does not support it. A small handful of dark-skinned grapes in the evening, Concord, Black Muscat, or dark red varieties, whole with the skins on. Not juiced, absorption is enhanced in a post-meal state, which is why the evening timing matters here. The 6th food is one I would not have included on this list 5 years ago, because 5 years ago, the specific compound it contains had not yet been linked to the senescence pathway in the published literature. Whole oats. And the compound is called avenanthramide. Avenanthramides are polyphenols found only in oats and in no other grain. They are potent inhibitors of NF-κB, the same transcription factor that allicin suppresses. But avenanthramides target a different upstream entry point, the IKB kinase complex. Think of it this way. Combining oats with crushed garlic gives you NF-κB suppression from 2 independent biochemical directions simultaneously. The redundancy is meaningful. It makes the suppression more consistent and more complete than either compound achieves alone. In 2021, the Journal of Gerontology published an 8-week human intervention study. 67 participants aged 55 to 72. Whole oats versus refined grain controls. The oat group showed statistically significant reductions in NF-κB activity, Illinois 6, and CRP. The effect was strongest at week 6, which tells us something important. The benefit accumulates with sustained use, not overnight. This is a compound that works in the background, week after week, until the inflammatory environment in your tissue is meaningfully different than it was before. Half a cup of whole rolled oats per day, not instant, not steel cut for this specific purpose. Rolled oats cooked in water. Instant oats have been heat-treated in ways that partially degrade the avenanthromide fraction. Before the protocol, take 2 seconds to check that you are subscribed. This week, I am publishing 4 more videos. Each one covers a different mechanism of cellular aging that your doctor almost certainly has not explained. This is the foundation. The rest of the week builds on it. At the start of this video, I promised you the cycling protocol, and I told you there is one detail in it that almost no one who covers this topic discusses. Here it is. The Mayo Clinic team found that the senolytic effect of fisetin and quercetin is dramatically enhanced by cycling. Not by daily use, continuous daily exposure appears to downregulate the senescent cell's sensitivity to these compounds over time. The cells adapt, but intermittent, concentrated exposure, 2 days per month at a higher dose, preserves and amplifies the apoptotic signal. The cells do not have time to adapt between cycles. Think of it this way. You do not run a controlled burn every day. You do it strategically, on a schedule, to clear the undergrowth before it becomes the fuel for something much larger and much harder to contain. This is the protocol that produced measurable reductions in biopsied human tissue in 2019. The gap between that published result and what any physician is currently recommending in a standard appointment is approximately 15 years. I do not think you have 15 years to wait. And the last thing I promised you at the beginning of this video, I saved it for right now. Because without the 6 foods you have just learned and the cycling framework you now understand, this final piece would not have made sense. This is the variable that determines whether the entire protocol works or whether you do everything right and still see no result. It is not a food. It is something most of you do at least once a week without thinking. And on cycling days, it has to stop 2 days per month. On consecutive days, in the morning before you eat anything, consume 20 fresh strawberries and 1 tablespoon of rinsed capers. Whole strawberries, not blended. Capers eaten cold. Nothing else for 90 minutes after. The fasted state is not optional. Fisetin and quercetin compete with dietary glucose for intestinal transport proteins. Taking them fasted increases absorption by approximately 30% compared to a post-meal state. This is the variable most people get wrong. Pick the same 2 days every month. The first and second of the month works as a simple anchor that requires no tracking. For the remaining 28 days, here is the maintenance phase. Half a raw red onion per day, sliced thin, with olive oil. 1 tablespoon of cold rinsed capers. 2 cloves of crushed garlic with 10 minutes of waiting time before eating, every single day. Half a cup of whole rolled oats at any meal. A small handful of dark-skinned grapes in the evening, and the most important part, 15 minutes of light walking within 1 hour of the cycling phase intake. A 2020 study published in Aging Cell demonstrated that moderate movement increases lymphatic circulation. This accelerates the clearance of SASP proteins from interstitial tissue. The senolytic foods reduce the cells. The walking helps purge what those cells were producing. Without those 15 minutes, you are leaving a significant portion of the protocol's effect behind, and the red line. This is the piece I saved for last. Ethanol competes with fisetin for cytochrome P450 metabolism. Clinical studies showed that it blocks the senolytic effect almost entirely within 30 minutes of intake, not reduces, blocks. 1 glass of wine on a cycling day negates the protocol, not partially, almost entirely. 30 minutes is all it takes. Cycling days are alcohol-free days. That is not a preference. That is the line between this working and this not working at all. If what you heard today is different from what your doctor has told you, that is not an accident. This is research that has not reached clinical practice yet. The gap is real, and closing that gap for women and men over 60 is exactly why this channel exists. Subscribe. Drop your age in the comments below. I read every single one, and tell me, had you ever heard of senescent cells before today? Tomorrow's video is about something most people have never thought about. You have a second circulatory system in your body, not the one that pumps blood. This one carries only waste, the debris from every immune battle your body fights every single day. Johns Hopkins published data in 2020 showing that after the age of 55, this system slows down by 40% in most adults. When it slows, immune waste accumulates around your organs, and dormant cancer cells that your immune system had contained begin finding the environment they need to escape. I am going to show you the one evening drink that the research says begins reversing this within 48 hours. That is tomorrow. It is on the screen right now. Do not miss it.